Alright, so today I'll be going over mitosis. If you want to learn more about meiosis, come to the workshop or come to my office hours. So the first phase of mitosis or stage is interphase. Okay, so in interphase, let's say that my cell has 3 different types of chromosomes, represented by my 3 lovely colors. In interphase, these are all duplicated. So we're going to have another red, we're going to make another green, and we're going to make a copy of another blue. And they all make these little X shapes, and they have centromeres attaching them at the bottom. So there would be 3 pairs of chromosomes and 6 centromeres at this phase. From interphase, we are going to go to prophase. In prophase, and in interphase, this is all in the nucleus. In prophase, I'll draw my nucleus a little bit smaller here. The nuclear envelope begins to kind of dissolve and disintegrate, so I'm going to represent that with a dashed line. And our centrosomes, are going to begin to create spindles, and we still have our 3 chromosomes in here. So this is what prophase looks like. The cell's beginning to get ready for replication, the nuclear envelope's disintegrating, and the centromeres, sorry, I lost my train of thought. Centromeres are beginning to create their spindle fibers. Our next phase is metaphase. So what happens is, this is pretty much a major reorganization of the cell. The chromosomes line up in the middle. And our centrosomes are on opposite sides. They're made up of 2 barrel-shaped centrioles. And what they do is they have a spindle fiber that attaches to each centromere of each chromatid. So if you remember from earlier in this section, these are microtubules and these are microtubular organization centers. So the spindle fibers are microtubules. From metaphase, we'll go to the next phase, which is anaphase. So in anaphase, what happens is the chromosomes are being pulled apart into their respective chromatids. And then we have our centrosome and our spindle fibers pulling our chromosomes apart. So this way we now see that there's the identical DNA going to each, what will eventually be a cell, but until then it's still being pulled apart. After anaphase is telophase. And cytokinesis. And to have a more accurate representation, I should probably erase this circle and instead draw a peanut-like thing. So what happens here is The chromosomes and now chromatids are separated into the formation of the two new cells. Nuclear envelopes are starting to reform. We have our centrioles on each side. And if you guys remember, I'll draw this in green. In the center we have plasma membrane pinching, and that is the actin microfilament and myosin walking around to pull it tighter and tighter and tighter until they are split. So what you will get is, in the end of it all, is you get our two daughter cells. They have their nucleus, their centrosomes, and of course all their other organelles that I don't really want to draw. And now the DNA is, instead of having our three separate chromatids, it's all back to being unorganized and a blob. And then through the right information to the cell and the right signaling, and when the cells are healthy, mature to grow, they'll go back into interphase and start the whole cycle over again.